Okay, let's dive into this. Welcome to today's Explainer. We're tackling a true medical mystery today, one that sits right at the intersection of our diets, our DNA, and that microscopic world inside our gut. We're talking about celiac disease and non-celiac gluten sensitivity. Now, both of these conditions share some really frustrating overlaps in how our bodies react to dietary gluten. But today we're going to zero in on a fascinating biological paradox. Think about this. Why is it that millions of people walk around carrying the genetic loaded gun for the autoimmune destruction of celiac disease, yet only a fraction actually have that trigger pulled? It's wild, right? To solve this, we're going to have to look closely at the systemic symptoms, the genetic suspects, and something incredibly intriguing known as the dark genome. To put things into perspective, around 1 in 133 people right here in the United States have celiac disease. Globally, it sits at about 1% of the population. But here's the thing. That number is actually creeping up over time. It's not just some dietary fad. This is a chronic, highly complex autoimmune condition. And if a strict diet isn't maintained, it comes with some really severe acute and long-term complications.
So what's actually happening at the baseline here? Well, when someone eats gluten, that protein found in cereals like wheat, barley, and rye, it gets digested into these smaller proteins called gliadins. These gliadins then get absorbed by the small intestine. But in susceptible individuals, this totally normal act of eating induces an autoimmune-like reaction. The real mastermind behind this is a specific enzyme called tissue transglutaminase 2, or TG2 for short. TG2 actually modifies that gluten peptide, essentially putting a target on its back. It makes it recognizable as a threat by the body's immune system, which sparks this abnormal T-cell response. And boom, that is the spark that lights the fire of the disease. Now, what's really interesting here is how this completely dismantles the myth that celiac disease or severe gluten sensitivity is just an upset stomach. While gastrointestinal issues like diarrhea, bloating, and unexplained weight loss are the classic hallmarks we all think of, the extraintestinal systemic symptoms are just vast. We're talking about aphthous ulcers in the mouth, a really severe skin rash called dermatitis herpetiformis, idiopathic neuropathies that literally affect the nervous system, muscle weakness, and even profound malnutrition leading to anemia. This disease radiates way beyond the gut, severely impacting the skin, the nervous system, and our blood vessels. It is a full body experience. To understand how a disease triggered in your stomach causes all those systemic, whole-body symptoms, we have to look at the physical destruction happening at the microscopic level. This gluten-triggered reaction physically destroys the gut lining. We're talking about villus atrophy, which is the literal flattening of those tiny finger-like projections in your intestine that absorb nutrients, along with something called crypthyperplasia. Because these vital structures are destroyed, the intestine simply cannot absorb nutrients properly. And that localized microscopic malabsorption, that is exactly the origin point for those systemic symptoms we just talked about, like bone disease and anemia. It's a domino effect. Of course, none of this chaos happens without the right genetic backdrop. The best understood genetic factors are the HLA variants, specifically high-risk variants of HLA-DQA1 and HLA-DQB1, along with the HLA-DQ2 or HLA-DQ8 genotypes. These genes encode cell surface proteins that are absolutely crucial for immune system function, and having them is widely recognized as mandatory for celiac to develop. But here's the catch. Geneticists calculate that these HLA variants account for only up to 40% of the disease's heritability. Genome-wide studies have spotted 39 distinct loci and 57 total single nucleotide polymorphisms tied to immune function that also play a role. Yet, even when you combine all of them, the currently identified genes only account for about half of the total genetic predisposition. So, we're missing half the puzzle. Which brings us to the core paradox of our explainer today. We know that the presence of these specific HLA immune genes in the general population is around 30 to 40%. Basically, about a third of all people have these so-called celiac genes. But only 1% of people actually become gluten intolerant and develop the disease. If the genes are required, why aren't they sufficient on their own? What is the missing trigger that separates the 33% who carry the risk from the 1% who actually suffer the autoimmune destruction? It's a massive medical mystery. To solve this paradox, researchers had to look way beyond our standard genetic coding and venture into what is mysteriously known as the dark genome. Dr. Ghosh from Columbia University provided a massive missing puzzle piece here. They discovered that it's not just about having the HLA-DQ2 or DQ8 genes. It's actually about a specific long, non-coding RNA known as LNC13. If you have the celiac genes coupled with a defective LNC13 gene, your likelihood of developing the disease just skyrockets.
This incredible finding proves that it's the inherited traits regulated by epigenetic modifications, so factors completely outside the primary DNA sequence, that finally pulled a trigger. And this brilliantly illustrates how the epigenetic switch actually works. It operates through environmental triggers, like dietary gluten exposure itself, or even changes in your intestinal microbiota. This trigger influences epigenetic regulation enzymes, which leads to DNA methylation. Basically, in active celiac patients, we see these distinct methylation profiles, particularly in the core NFKB pathway, which is absolutely crucial for immune cellular functions. Genes associated with this pathway, like MLT1 and MAP3K7, become hypermethylated in the small intestine. It essentially flips the switch, upregulating the pathway and activating the full-blown autoimmune disease. It's like turning the volume dial up to 11. Now, these hypermethylated, hyperactive pathways aren't just theoretical. They are highly visible in transcriptomic profiling. We literally see a massive upregulation in the immune system, cytokine signaling, and interferon signaling pathways, specifically interferon gamma and alpha/beta signaling. This is the physical reality of what happens when those epigenetic switches are flipped. The mucosal transcriptome is fundamentally altered, driving those abnormal T-cell responses and all the inflammation that ultimately destroys the gut lining. So the absolutely crucial point here is how this molecular understanding completely changes how we view treatment. Right now, the only accepted treatment is a lifelong, strict gluten-free diet. And hey, on the surface, the clinical appearance is great. Patients become symptom-free and their gut mucosa visibly heals. But the transcriptomic reality reveals a totally different story. Even on a strict gluten-free diet, patients still display patterns of ongoing disease at the molecular level. The data shows hyperactive Wnt signaling, which results in immature crypt gene expression. So it turns out that patients on a gluten-free diet are not completely healthy at a cellular level. Their cellular differentiation is still impeded by these deep molecular alterations. They're healed on the outside, but not quite on the inside. Which leaves us with a final, rather profound question. We've journeyed all the way from recognizing systemic, whole-body symptoms down to the genetic loci, and finally into the hidden switches of the dark genome. We now know that simply removing gluten heals the surface, but molecularly, the immune and cellular pathways remain altered. So how will unraveling these specific epigenetic profiles, like DNA methylation and non-coding RNAs, lead us from a simple diet to a true molecular cure? As we integrate machine learning and transcriptomics, we're standing right on the edge of finding entirely novel therapeutic approaches. The future of autoimmune treatment won't just be about avoiding a protein in our food. It will be about rewriting the epigenetic code itself. Thank you for joining me for this explainer, and I hope this sparked your curiosity to learn even more about the incredible, fascinating world inside ourselves.